Welcome viewers. Today I am going to discuss about advanced microscopic techniques, and we will talk about phase contrast microscope, polarized light microscope, differential interference contrast or Nomarski microscope, and fluorescence microscope. And fluorescence microscope can be categorized into wide field microscope, total internal reflection fluorescence microscope, and confocal microscope. Now coming to phase contrast microscope, as the name tells that we want change in phase of light to enhance the contrast. The optics of it was first devised in 1930s by Fritz Zernike, who suggested that if light passes through an object, depending on the thickness of the object, there can be a change in the phase of the light compared to light which is passing through the background. This is amplified by a few optical elements in the instrument and is reflected as difference in intensities of light which our eye can detect as due to change in contrast. Now, going to the details of phase contrast microscope, first we should understand where does this phase difference comes from in biological specimens. In biological specimens, depending on its thickness, the light traverses through the specimen as well as through the background. Now we know that the velocity of light depends on the velocity of light in vacuum by the refractive index of medium. So a thicker medium, the velocity of light will decrease and correspondingly there will be more deviations also. Now the objects which can actually change the phase of the light ray passing through it are known as the phase object. As light passes through the specimen, it is then collected by the objective which focuses into the next elements, which may be the ocular lenses, our eyes, or the camera. Now, it should be mentioned that this difference of the 2 points, that is light passing from the object versus the background, leads to what is known as the phase shift. As you see in the figure, the phase shift, which is small delta, which is equal to 2 pi delta by lambda, where capital delta is known as the optical path difference. Therefore, the phase shift, which is equal to 2 pi capital delta by lambda, where lambda is the wavelength of light, delta is the optical path difference.
Sir, what do you mean by optical path difference?
Optical path difference is the difference of path length of light traversing through the specimen and the surroundings. And this depends on the difference of the refractive index of the specimen versus surrounding as well as the thickness. Now coming to another important concept in phase contrast microscope is the light traversing through it. Now as the incident light traverses through the condenser lens and then through the object, it is broken up into 2 fragments. One which gets deviated due to interaction with the specimen or the object. This is known as the diffracted ray or D ray, and another which pass undeviated, which is known as the surround ray or the S ray. Both these rays next crosses through the other objects of the instruments and then they interact with each other. After they interact with each other, the wave which is given rise to is known as the particle wave or the P wave. So as you see in the equation, P wave is equal to S plus D. Now how is the phase difference created, or how is the phase shift happening? Now if you look at the figure, as the light comes out through an annulus, which is an important component of phase contrast microscope, it is focused into the object or the specimen, and then from the specimen it gets into the phase ring. Now while it goes through the specimen, the D ray is actually decreased by lambda by 4 compared to the S ray. Now a phase plate is an important component of the phase contrast microscope, and this actually enhance the phase of the S wave by lambda by 4. So if you now think of it with the object as well as the phase plate, the S wave is enhanced by lambda by 2 compared to the D wave. Now when these 2 waves now meet, they will cause destructive interference and this interference pattern is what is reflected as intensity changes, due to which we can actually see the surfaces or even the boundaries of the cell distinctly from its surrounding. Now, phase plates can be of two types, positive and negative.
Sir, what are positive and negative phase plates? Is there any difference in the design of them?
Yes, there's a distinct difference in the design of the positive and negative phase plates. If you look at the figure, a positive phase plate has a ring of etched glass, due to which the S wave is advanced by λ/4, while in case of a negative phase plate, because of the design, the S wave is retarded by λ/4. And the advantage, so objects with low refractive indexes appear brighter if you use a positive phase plate. On the other hand, if you use a negative phase plate, objects with high refractive index appear brighter. Now, phase contrast microscope is widely used in biology to see objects without staining. So nowadays, cell cultures or other tissue cultures are observed under phase contrast microscope. Even the movement of different cells, the extension of the filopodia or the lamellopodia can distinctly be observed using phase contrast microscopy. The next type of microscopy which I am going to discuss is polarized light microscopy. As the name suggests, that polarized light is used to illuminate objects. And what is the importance of polarized light and what is polarized light? Polarized light is a light where the electrical vectors vibrate in a particular direction compared to unpolarized or non-polarized light, a good example of which is sunlight. Now, this electrical vectors, when it vibrates in one direction, when such light interacts with the birefringent object, which are objects where there is some difference in two phases, so that the refraction or the deviation from one phase is not the same as deviation from the other phase, then the electrical vectors will be made to vibrate in some other direction, and hence from such measurements we can know about the nature of birefringence of these objects. So in polarized light microscopy, the use of polarized light is because of this particular reason. Now, what is done in such type of microscope is that when light, as you see in the figure, when light passes from the source, it then passes through a polarizer, which is a ground glass having multiple coating, which leads to polarization of light. This polarized light now passes through the condenser, focused onto the specimen, passes through the specimen, and then whatever is coming out from the specimen is collected by the objective, and on the backfocal plane of the objective there's an analyzer. Now, the importance of using of these polarized objective is that they're not the same as the objective which are used in other bright field microscope. These objectives are polarized objective, especially ground glasses, where the stress or the shear is very less, and these are known as PO lenses, P lenses, or Paul lenses. Now, once the light passes through the objective, it will pass through the analyzer and then is collected to the eyepiece or to the camera. I should mention here that the analyzer and polarizer are placed in a crossed position. So what I mean by that if this is how the polarizer is placed, the analyzer will be placed like that is 90 degree to each other. Because of which, if light is vibrating in one direction and it passes through the polarizer, the analyzer will prevent that light to pass through. However, if you put an object in between that, and if the object is anisotropic in nature.
Sir, what do you mean by anisotropy and how can it be observed?
Yeah, anisotropy as opposed to isotropy is the difference of light refraction of two faces of a particular object. The best example is the lipid bilayer. Now, as you know, the lipid bilayer has proteins in it. Now, the proteins or the arrangement of proteins on the outer leaflet of that bilayer is not the same as the inner leaflet. Because of that, it acts as a perfect example of an anisotropic membrane. Now, as light goes through one membrane or the outer leaflet, and it will be refracted in a manner which is not the same of refraction as light comes through the inner leaflet. And this anisotropy can be well studied using polarized light microscopy. Now, so after light passes through such an anisotropic object, depending on the nature of anisotropy, the polarization of light will change, and this light will then pass through an analyzer, and in certain cases there can be the Bartrand lenses, which are projector lenses which project such images and takes it to the computer. Polarized light microscope is used to check the anisotropy present in the membrane, or even study the dynamics of different proteins in different leaflet of the membrane, as well as it can be used not only in biology but also in mineralogy, where it was actually essentially first used. Now, coming to the third type of microscope, it is known as differential interference contrast microscope. Now, this microscope has subtle changes from what we have studied in the phase contrast microscope. One of the problems of phase contrast microscope are phase halos and shadows, which can be dealt with in a better way in DIC or Nomarsky microscope. It was in 1955 when this design of the microscope was first propounded, and later it was worked upon by George Nomarsky, because of which it is known as Nomarsky optics. Now, how does it work? What are the components? If you look at the figure, you will see that light, after coming from the source, unlike the polarized, it will now pass through a modified Wollaston prism, which is also known as the Nomarsky prism. Now, what it does is break up the light into two rays, and the distance between these two rays is known as the shear.
So what is the importance of shear distance?
Yeah, that's a very good question. Now, shear distance is the distance between these two rays, and one should always keep in mind that this shear distance should be less than the resolving power of the objective. Otherwise, a double image will be formed. Now, once it passes through the object, these are collected depending on the interaction of this light ray, both the light ray with the object, it will come to the objective. I should hereby mention, of the two rays, the design is such that one will pass the object, the other acts as the reference wave. Now, once it passes to the objective, then there's a second Wollaston prism which actually joins these two rays, because of this the shear is gone. This creates an interference pattern, and it is this interference pattern which will help to us to visualize this object, because it will increase the contrast of the object. An important feature of DIC microscope compared to phase contrast microscope is that because of this traversing of the light ray through the object and because of the reference beam, sometimes there's a pseudo 3D appearance happening of the objective. So the objects look as if a 3D object. But this actually happens because there is a very thickness in the sample, and DIC can utilize this thickness which cannot be utilized or which cannot be observed in phase contrast microscopes. A fluorescent microscope uses xenon or mercury arc lamp. The reason of using this is we can obtain specific bands of wavelength from these lamps. For example, for mercury arc lamp, we can obtain anything from around 350 to around 570 nanometers. However, in modern day, for specific illumination, what people use are lasers. Now, the importance of use of lasers is that By using a laser, I can have a fixed line or particular bands or particular spectral wavelength coming through to illuminate the specimen. Now, different lasers are there. For example, argon ion lasers, which has to be air cooled, are used for illumination in the UV range. For illumination in the green range or in the red range or in the far red range, Hini lasers are used. Now, the next important component of the microscope are the filters. Now, filters are mostly three types. The first filter which people come across are the excitation filter, next the dichroic mirror, and the third the emission filter. You can see the arrangement looking at the figure. Now, the excitation filters are usually band pass filter which allow a particular band of wavelength or light to pass through. So let's say I want light of only coming around 470 to 480 nanometer or even 490. So the excitation filter has to be adjusted only to allow that band of light. Now, after the light passes through it passes through a dichroic mirror. The importance of a dichroic mirror is that the glass or the optics is such that it allows shorter wavelength to get reflected while the longer wavelength could pass through. As you know, in fluorescence, the incident light are shorter wavelength than the emitted light. So the emitted light will pass through the dichroic mirror. It is arranged at an angle of 45 degree in the filter cube. After it passes through the dichroic mirror, the emitted light or the fluorescent light passes through the emission filter, which cuts off all other light except that emitted light, and then it passes through the detecting devices or to our eye. Now, before going into the divisions of fluorescence microscope, I want to make you aware of two important concepts which lead to loss in fluorescence. They are quenching and photobleaching. While quenching is a reversible phenomena, and this can happen due to interaction of the fluorophore with different components of the medium in a cell culture or with different ions, resulting in lessening of the fluorescence intensity. Photobleaching is an irreversible phenomena which can happen if, let's say, you expose the sample for intense laser for a long duration of time. Now, coming to the different types of microscope, first there is wide field fluorescence microscope. So what is that? This is just an advanced stage of a normal microscope or the microscope we have used in phase contrast or even in bright field microscope with the filter set and the mercury arc lamp or the laser attached to it. Now, wide field microscope is widely used in biology to visualize different cells. It can actually eliminate all the fluorophores which are present in the cell. However, it has its own fallacies. If we want to see particular fluorophore at a particular region of the cell, wide field microscope is a major drawback. Now, because of this, different types of modification of fluorescence microscope has been done. The first is TIR-FM or total internal reflection fluorescent microscope, which is also known as the evanescent wave microscope.
Sir, how is evanescent wave generated?
Yeah, to understand TIR-FM, I think the first thing we should understand is the generation of evanescent wave. Now, we know the concept of refraction. So let's say when light passes from a optically denser medium to rarer medium, the lights get deviated. Now, if we attain the critical angle, then light will not go to the second medium. And if we go more than the critical angle, most of the light will be totally reflected. That is the phenomena of total internal reflection. What we have not heard before in our school days is that at the point of incidence, while the total internal reflection has happened, a ray is even emitted. And this ray is known as the evanescent ray. So if this is how the light is coming and getting reflected, from this point a ray is emanated in 90 degree to this direction of propagation. So let's say this is glass, evanescent wave will be generated and come into this side. And so cells which are present on this side will be illuminated. Now, how much of the cell will be illuminated? As you look into the figure, the evanescent wave is a fast decaying wave with a very less penetration depth. So at least 100 to 200 nanometer of thickness can be illuminated by evanescent wave, and which also suggests that not the total of cell can be illuminated by such a wave. Only near membrane events or fluorophores which are present near the membrane can be observed, and that's the crux of evanescent field microscopy or total internal reflection fluorescence microscopy, where without illuminating the whole cell, we just observed the dynamics of different proteins tagged to fluorophore at the plasma membrane or near the plasma membrane. Even trafficking of various proteins or movement of vesicles which are attached with fluorophores can be visualized.
So, is there any optical method to look at specific organelle within the cell?
Yeah, there are optical methods to look at specific organelles. For example, if I want to look at the Golgi apparatus or fluorophores or proteins which are present on the Golgi apparatus, or let's say on a particular vesicle inside the cytosol, without looking at the cell as a whole, then we can use confocal microscope. And that's actually the advantage of using a confocal microscope. Now, confocal microscope can be categorized into two types. One is laser scanning confocal microscope, and second, tandem scanning confocal microscope. What is laser scanning confocal microscope? Now, the basis of confocal is the presence of pinhole. So, after the light source, which is in most cases the lasers or the mercury arc lamp, once it passes through the excitation filter, it crosses a pinhole. The pinhole is such adjusted so that the light is focused to a particular region of the specimen or the object. After passing through the object, the light can be refracted or slightly deviated, and the fluorescence light which is obtained is collected by the objective. This objective now passes it through an emission filter, and then it passes through another pinhole. Now, why is the importance of the second pinhole? The second pinholes actually cuts off all the out of focus light which can be refracted or deflected that I told you, and only collect the focused light or the focused fluorescence light which is coming from the object. Then after this is collected, it is taken through a camera, which in most cases is a PMT or a photo multiplier tube, which converts the language of the photons into electrons, amplify it, and then by another device is converted to digital signal and can be transferred to a computer. So confocal is a very good example of an integrated microscopic system where using computer software I can actually operate the microscope. I can also control the position of the pinholes and the movement of the pinholes. I should also tell, in case of laser scanning confocal microscope, mirrors are there which reflect light coming from the first pinholes, and what it does is, if this is the surface of the sample, light first impinges here and then a raster scan happens. So it's a zigzag scan so that all XY of the specimen is sampled. Then, depending on the position of the second pinhole, it moves to the next layer, and similarly raster scanning of the second layer happens. The implication of raster scan and collection of information from all layers is that once we have the whole information of the cell, we also have information from all the layers. And this method is known as optical sectioning. That is, without physically sectioning the sample, we have information from all layers of the sample. That Z distance, that is the difference between these two layers, can also be controlled. Okay. Now, another important implication of confocal microscope is that because of the arrangement of the filters and the pinholes, and because of use of lasers, we can use more than one fluorophore and image them in one go. However, there's a major problem, that is the problem of bleed through.
So what is bleed through?
Yeah, bleed through, as I was discussing, is coming of light from one channel to another. Let's say, and this happens especially for simultaneous scanning of more than one fluorophore. So let's say filters were such arranged that blue light emitted by a fluorophore should have come from filters of the blue, and green light should have come to the filters of the green. However, somehow the blue light comes into the filters of the green or the green channel. This is a good example of bleed-through. In most cases in optical devices, 10% bleed-through can still happen. However, nowadays glasses or the filter settings are so made that bleed-through is minimal. Another way of evading this bleed-through is taking sequential scan. However, the problem with sequential scan is that while scanning for one fluorophore, the other fluorophore may also be affected. Now, because of this, another type of microscope, which is a confocal microscope, which is used, is known as the tandem scanning confocal microscope, in which instead of raster scan, there's a NIPCO disc where there are small holes or pinholes along with the micro lenses. Now, it can scan the specimen in one go, due to which there's an increase in speed of the scanning, as well as there is an increase in spatial resolution, better signal to noise ratio. However, the drawback of NIPCO disc microscope or tandem scanning microscopy is that the micro lenses in that rotating disc has some glass or optics where UV light is not best utilized. Now, what both these microscopy, that is the laser scanning as well as the tandem scanning, can be used to see various features of a cell, to even do live imaging and also observe co-localization, that is localization of two proteins in the cell, and it can also be seen the interaction of two proteins by using two fluorophores. So coming to the concluding section of this lecture, what we have discussed today is phase contrast, polarizing and DIC microscopy, which can be utilized to visualize the cells without staining them. Again, if we use fluorophore, we can see various parts of a cell or the cell in entity or even the near membrane events by different types of fluorescence microscopy. Thank you very much.